Hello, my name's TJ Martin. Today I'm going to be talking to you guys about the chemical origins of fruity and savory exotic cannabis aromas. And so why are we looking into this, and why should we all care about why cannabis smells and tastes the way it does? Well, a lot of when consumers are going to a dispensary and they're making their purchasing decisions, a lot of it may be based on THC content, obviously a very well-known quantity in the industry. Bag appeal, especially when you're thinking about things like how the flower bud itself looks, or when you're looking at concentrates, what's the color of the concentrate and the consistency. But another thing to consider is also how does it smell and taste, right? When a consumer takes this product home, they are going to smoke and consume it. They have some predisposition or some thought about what it's going to smell or taste like when they bring it home. And if it's something they enjoy, it's going to be something that may drive these decisions for why they decide to repurchase said products. And the aroma of cannabis has this broad range, right? It can range anything from gassy and sweet, woody and citrus, spicy, creamy, even things such as grape, tropical, gassy citrus, skunky, chemical, or even more nuanced things like papayas and pineapples. And so what is some of the chemistry that we know right now? A lot of it, and this is a Sankey diagram. What a Sankey diagram does is it breaks down the metabolites of interest into how they are based on weight. And so these are proportional to the weight that's inside the actual cannabis flower material. Obviously, cannabinoids take up a large portion of it, specifically THCA, because what I'm looking at today is high THC cannabis. But then also breaks down into other things like other class, which would be flavonoids, fats, waxes, and aroma compounds. In aroma compounds, you break down into things like terpenes or flavorants. Terpenes are a subclass of flavorants, but for the purposes of this, I will keep them separate. And flavorants break down into esters, alcohols, aldehydes, sulfur compounds. And these are some of the lowest weight content materials in the plant, but they may have some of the highest odor impacts to the actual material itself. And so the aromatic categorization of cannabis as we know it today, obviously you have prototypical, that's your traditional OG Kushes, things like wedding cake, Gorilla Glue are very spicy, herbal and gassy, as well as Jack Herrers and hazes that have some citrus notes to them, very piney, very terpenic. But when you start thinking outside of the classical range, you start thinking things about like sweet exotics and savory exotics. So when we started this, we really wanted to understand what makes sweet exotic cannabis smell and taste the way it does. Obviously, we know a lot about terpenes. Terpenes are in the plant, those have been studied heavily, but what are the correlations in the plant that lead to make a strain? Like Zittles have some of its unique candy notes. Some more exotic berry cannabis like Blue Icy, Blackberry Kush have their dark berry notes. And as we were going through this, we were running some our sensory panels internally and giving people free questionnaires like, hey, can you please describe this cannabis in as little terms as you can, be open and honest about it. We were originally starting to look at just how does sweet differentiate from prototypical, and we were using actually garlic cookies as a control. And one of our panelists, we asked them like, hey, would you consider this a exotic or not? And they said, well, it's not a sweet exotic, but in the context of is it prototypical? No, not really either, right? It's more of a savory exotic. And so as we started going through this, we're wondering like, hey, is there chemistry behind this that explains how there's also savory exotic in cannabis? And as you start seeing the trends in the industry today, we're seeing things like Donny Burger and pot roast coming in more popularity, the meat breath strains, right? Obviously funny name, right? But that's just how the industry goes. But these are more nuanced strains that are definitely not sweet, but they're also not prototypical either. So there's got to be some chemistry there that can explain those differences. And so we started everything from specifically rosin in this study. The reason we used rosin was it allows us to get a higher concentration of the volatile flavorants than flour. Flour has a range of 0.5% to 3 or 4%. Depending on the specific cultivar of choice, whereas rosin can vary from 5 to 15%, depending on the cultivar and how it's extracted. It also allows us to get a homogeneous sample going onto our GC. When someone makes rosin, they're using multiple plants that are going through its extraction process. If you take just a singular flower bud, you're pulling it from just one plant. Depending on where you pull it from the plant, it may have different impacts on the aroma. This allows us to get a homogeneous sample. It also has minimal processing. We're not worried about solvent carryover or solvent contributions to anything. It just strictly allows us to get what is truest to the sample with as minimal processing and most homogeneity possible. And so some of the data that came through our initial sensory panels, we were running through this. We have on top our 4 most sweet varieties, and on bottom our 4 most savory varieties. As you can see on what we have here, our density plots, this allows us to demonstrate how much some of these terms were used to describe these strains. So things like grape pie do-si-do had a lot of sweet, fruity, dark berry notes claimed, and things like papaya peach and Juice Man had a lot of sweet, fruity, as well as some tropical and citrus notes being claimed, but not very many savory notes. Whereas the savory varieties had a lot of these savory, olaceous, sulfuric, chemical notes coming up. And the reason we did this is with this study, we really wanted to correlate trends in sensory data to actual hard chemical data on the samples that we were measuring. And see if we can start to find certain compounds that are highly responsible for why these strains smell and taste the way they do. And if you look, something you'll notice as well is that all of these all have the prototypical aroma of cannabis always noted within these strains. There are slight variations from cultivar to cultivar, but it is always there, which is really important to note. And one of the first things we wanted to know is any of the exotic aroma due to specifically the main terpenes that are present, the major weight compounds? And so what we plot here on the left in figure A is we have exotic score from 0 to 100, how those ranked by the panel, as well as where that ranked in relation to one another. And as you look through the plot, there is no real trend between any of the major terpenes. It's kind of all over the board. Same thing on the right when we try and trend, is there any specific trends with the total number of aroma classifiers? In the context of sweet exotic, there was. When it gets more of a sweet exotic, it gets a lot to the consumers, it's more nuanced, it gets more difficult to explain, so you get a lot more descriptors, versus savory and prototypical, there's not as much correlation. So we went ahead and looked at however all these correlated to one another. So what we have here is a correlation matrix and from decreasing exotic score from the top to the bottom. And what this plot allows us to do is compare each cultivar to one another from all their sensory descriptors and say, hey, how are these similar or not? Are there any relationships between each cultivars? And so the lighter the color, the more similar they are. The darker the color, the less similar they are. And we noticed there was a huge diversity here, which was great. It's what we wanted. We wanted a lot of diversity because that allows us to then start looking at the chemistry, find trends, and start to understand more about this complex field of cannabis science. Something was interesting. We saw Gorilla Glue this specific sample was very similar to garlic cocktail number 7. I'll come back to that a little bit later, but it's important to note that these should not really have much similarity, to be honest. So now we have our sensory data. Now we can go in and start actually doing some of the hard chemistry. And what we're doing is we're using GC cross GC with a TOF mass spec detector and an FID. GC cross GC is a type of two-dimensional gas chromatography. What that means is it allows us to employ a traditional gas chromatograph where you separate based on compound size, but then at the end it has a pulsed flow modulator which allows you to then separate on a second column in that same GC, but then by polarity. So you can get a greater degree of separation and you can see more of what's in these samples than what you'd originally have access to. And as we were going through this, we built out a custom standards for all of our quantitation. We worked with different standards companies to get as many compounds as commercially available in an actual calibration standard, so these can be based on calibrated values and really try and find these trends. We're also using direct injection over headspace. This allows us to get as accurate and reproducible data as possible and get more mass on column and see more of those low analytes, because a lot of these, like I showed in the Sankey diagram, are very low weight amounts, so you have a high chance that you may not be able to see it if you don't get enough material on the GC itself. And this is the data we got. We ran through all 30 samples, ranking from super savory to classical to exotic, and everything pretty much looks the same when you look at a top-down view of it, because what you see on the GC is a lot of the major compounds which don't differentiate that much between cultivar to cultivar. And so when you look at it, it's like here we have grape pie docido, our most fruity exotic, and GMO, our most savory, and the chromatograms look almost identical. The monoterpene region looks very similar. The sesquiterpene later region looks almost identical. So I'm going to dive deeper into this. So what we know aromatically from sensory, these are different. They smell different, they taste different, but the GC data looks very similar, so it means there must be some minor compounds that are heavily contributing to these differences. And so we went back to our correlation matrices, but instead of looking at actually just the sensory differentiation, now let's actually do it on all analytes. So here on the left, we have all analytes detected for the samples, and we put it into a correlation matrix. And again, this is based on how light it is how similar they are, how dark it is, how different each strain is to one another. And you can see some differences, but not a ton, to be honest, other than bubblegum zittles, Gorilla Glue, and Trainwreck. Trainwreck obviously is a high terpenoid-based strain, which is one of the few terpenoid-based rosins we were able to actually find in Southern California for this study, so that one is going to be different. But if you go down and now break it down, and let's look at compare monoterpenes and monoterpenoids. It still looks pretty similar. You don't see a lot of heavy differences, again, showing that monoterpenes and monoterpenoids, while important to cannabis, aren't the differentiating factor in these exotic aromas. And if you go further and look at sesquiterpenes and sesquiterpenoids, they look almost identical. Like these are all 0.95, 0.99, or even 1.0 similarities, meaning they're 95, 99% similar. Now, if you take it a bit further and go just the flavorants, remove everything else out, move all the terpenes, monoterps, sesquiterps out, you see huge differences. This is where you start seeing a lot of the variation between these cultivars, and that plot now looks a lot more like our sensory correlation matrices. And when we started looking at data, we found is a whole bunch of different flavorants. And these are some of the key ones we wanted to call out because they are really important to these various aromas. So, and some of these are all well-known compounds by and large in the traditional flavor chemistry realm, but not necessarily in the context of cannabis and what it does in cannabis flavors and aromas. And so on like the fruity side, fruity floral, you have things like methylanthronyllate, really common like Concord grape, purple grape soda flavor. Some of the really interesting ones are the creamy sweet compounds, these like 6-amyl alpha pyrone, and some of these other compounds in that realm that are purple. These are very much your stone fruit, coconut flavors. Chemical naphthyl, these are heterocycles. We've never really seen nitrogen heterocycles in cannabis. Normally when we think about flavorants, we're thinking of carbon, oxygen, possibly sulfur. These are super interesting to see. Tropical citrus is a new class of tropical sulfur compounds in cannabis that they've been found other places, but these are driving some of these unique flavor notes that are in tangy strains, these weird citrus notes you don't get anywhere else. Fusel sweet, these are Senecioates. These are when you're think of strains like zittles, runts, gelatos, really heavily in zittles, to be honest. These compounds give it that what makes zittles unique and different from other exotics that are even berry notes is these Senecioate compounds. And in the fruity sweet, these ethyl butyrate, ethyl isovalerate, really important estery, very sharp top note that helps, again, push this aroma for cannabis. And so when you think about savory exotics, why do these GMOs, these garlic cookies have this unique scent to them? And so we found we were comparing our sensory data to our GC data, we noticed as there are some key non-terpene compounds that correlate with this intensely savory aroma. And these was Indole and Skatole. Skatole is found in human feces and mammalian feces. It gives poop its smell. And it has a wide range of aromatic impact. It's used heavily in the fragrance industry as well, because depending on the dose where you use it, it can impact things from what we know as savory, garlicky, and cannabis, all the way to just chemical naphthol, like mothball scent, and pretty off-putting, to be honest. But what's interesting is if you look at its chemical structure, it's actually very similar to what you see in melatonin. And this is, again, Indole and Skatole, pretty ubiquitous in nature. It's interesting to see it in cannabis. But to highlight just how difficult it is to actually see these, this is just, what we're looking here is GMO rosin, which is some of the richest in these compounds we've measured. On the top plot there, you have a linear scale. I've highlighted in the box with the red circles. You can't see them. Now, if you put it on a log scale and start really blowing it up, you can see it a little bit. You really have to zoom into the region and see that there's these tiny little peaks that are really buried in the chromatogram. And if this was on a one-dimensional GC, it would have been incredibly tough to find this unless you already knew they were there and built a method for detecting it. And so these were, again, kudos to the analytical chemists working on this, Dr. Ian Oswald, for finding these really buried in there. But these are so important to why GMO, ChemDog, these various strains smell and taste the way they do. And just another interesting piece about these, Skatole is very similar to 3,3-diindole methane. It's a CB2 receptor agonist. That specific compound has promising anti-cancer properties for certain cell lines. It also is found in various vegetables. What that compound is, it is two skatoles bonded together. So maybe Skatole has some impacts for different aspects of cannabis, whether it's the mood effects or medicinal properties. Then getting more into now the tropical VSCs, these other compounds that are being, when we looked in our data, correlated heavily with why do these tropical and petroleum citrus strains smell so unique and different from other strains. We notice is there is 3-mercaptohexanol, 3-mercaptohexylacetate, and 3-mercaptohexylbutyrate. Interestingly enough, this is the Gorilla Glue I mentioned earlier. It had similarities with garlic cocktail number 7. Garlic cocktail is a very petroleum citrus tropical strain. Gorilla Glue is not. Gorilla Glue actually, and when I went and procured the sample for the study, we went and got it as a control for our sensory panel to use because Gorilla Glue should be very spicy, herbal, some gassiness, earthiness. And we brought it in, I remember opening it in our sensory panel, and I smelled it, I was getting ready to do sensory, and just knew on the spot, like, this was not a real Gorilla Glue. Gorilla Glue does not smell tropical, and this was kind of sucked for the study because we were hoping to have a nice control, but it's also a positive as well, right? This is a good use case that can show how you can use the chemistry of cannabis to start authenticating are certain strains the ones that we say they are, right? The industry is well known to have renames, people borrowing genetics from one another. Things can be lost in translation over the years. But the chemistry doesn't change if the cultivar is what it is, and this shows very heavily so this Gorilla Glue is not a Gorilla Glue. It's very much a Tangy type because Gorilla Glue would not have these. And here are these are some of the Senecioates we identified in, like I said, some of the Indole strains. These are a new class of flavor compounds in cannabis. They have aromas that range from fruity, banana, pear, green apple, the very top note heavy. Some of these don't exist in other fruits, some do. These are also not commercially available, so we had to go in the lab and synthetically create, purify, and isolate all of these, and then put these through our sensory panel to see what do they smell like, right? Because I can't just go buy this online and bring it in and taste it, so we can only smell it, but what is their aromatic contributions? And this is really what helps differentiate Indoles differently from other exotic cultivars. It's also important to note that this is not present at all in OGs and other mildly exotics, and not these really intensely sweet fruity ones. It's just not there. I want to see is like just aroma percent and some of these specific major classes of compounds, do they dictate aroma quality? So we know they don't push the exotic notes of these, and it's not correlated to the exoticness of cannabis samples, but does it relate to the aromatic quality of the sensory panel? So we're relating the aromatic fraction weight and the exotic score, and you don't see, again, with these major terpenes, again, no correlation. Flavorants, you have this huge correlation. Monoterpenes, monoterpenoids, basically flatline sesquiterpenes, sesquiterpenoids actually decreases. Kind of makes sense. Sesquiterpenes, sesquiterpenoids are associated with older aged cannabis typically because these are some of the things that flash off the last and they volatilize the slowest rate. So there may be some negative connotation there for some panelists. And now looking, maybe it's just not just monoterpenes. Maybe there's a specific monoterpene that does drive some of these aromatic exotic aromas and the perceived exotic aromas. And so we looked at them all, and really there was none. They were all pretty much flat lines for the most part, indicating that, again, while these major monoterpenes are important in the context of cannabis, they don't differentiate the exotic nature of these various cultivars. So when to look in the bottom is like, well, maybe it's related to flavorants, maybe it's related to the tropical VSCs in the middle, or even the heterocycles like Indole and Skatole on the bottom right. And we noticed is that it is. That's what those heat plots are showing, is that as they get a higher concentration, we start seeing more of these compounds, it does trend with these compounds, which makes sense. As we were comparing the sensory data to the GC data we expect of these written, this just further confirms that notion. And so with this, now we've been able to expand our understanding of the exotic cannabis aroma spectrum and able to start fleshing out what is sweet exotic cannabis, what are the compounds responsible as fruity, berry, tropical, unique citrus notes, as well as on the savory side, what's responsible for those onion, garlic, chemical notes, and get a broader, more in-depth understanding of why exotic cannabis smells and tastes the way it does. And so some of the conclusions here, we notice terpenes minimally, if at all, influence the perceived exotic nature of cannabis. The small concentration compounds here, flavorants, are really combining to heavily impact the unique attributes. Skatole is one of the major components, with Indole impacting that chem, funk, savory, unique note, these elaceous note. Not really elaceous, but it gets perceived as such in these exotic savory strains. And then the tropical VSCs really impact those unique tangy citrus notes. Some of the future work we're looking into is looking at PCA and other statistical classification analyses, different formulating algorithms to accurately predict some of these aromatic properties. Can we relate these into more digestible metrics for consumers? Obviously, you're very sensitive and understand that this is very hard chemistry. These are very complex compound names. It doesn't really mean much to an end consumer. Putting you have 3-mercaptohexylacetate on a container at a high level means nothing to them. If anything, it's probably scary, right? But these are meaningful impacts to chemistry. There's probably a way we can use this in a meaningful way to educate consumers in maybe various ranking scores and systems, right? Or there's got to be another way to educate consumers and at least help them move away from just THC and realize there is other things at play driving these experiences that they so much love and enjoy. And then lastly, structural solution of other unknowns. There's lots of other unknowns in these cannabis cultivars that we haven't solved. Cannabis produces tons and tons of compounds. We're publishing on these as we find these, and we understand the unique chemistry that relates to the aromatic properties and what these relationships are, but there's still a lot more out there. And just one other thing I would like to mention on this whole thing is that this can be oftentimes perceived as terpenes are irrelevant in cannabis, which is not true, right? When this study was originally published, we saw a lot of articles out there take this and put it in the context of terpenes are irrelevant. They're not irrelevant, actually. Because when we did this and we created a lot of our different products, we stripped out all the terpenes and made blends with just the flavorants, just all the fruity flavorants, the chem notes, the thiols, everything except for the terpenes. And it has no semblance of cannabis. It doesn't smell terrible, but it doesn't smell like cannabis at all, because terpenes are the foundational piece to why it smells and tastes the way it does. It helps bridge and marry everything together. It's the right ratios, everything that can give you that symphony that is each cannabis aroma. With that, I'd like to acknowledge the team. Huge thanks to Dr. Ian Oswald, heavily led this study and found a lot of why these things smell and taste the way they do with me. Twinkle and Manny Marcos did a lot of the lab work, a lot of the data analysis with us. Manny did all the synthesis of the Senecioates. Kevin, our CSO, for always helping support and helping drive this research forward. Huge shout out to 710 Labs, great collaborator of ours. They provided a lot of high-end fresh rosin that as soon as it was done, that way we could have ideal samples. Something that isn't lost on us is that the fresher the sample, the better the data. So you want as fresh as you can possible, because then the GC can only detect so much. And then shout out to the rest of the entire abstracts team, Sepsolve Analytical, Marks International, for all the help and collaboration over the years. With that, I'll take questions.
Great presentation, thanks. The question I have is about the the shelf life and delicacy of some of these things you're finding, these esters. I've noticed from cultivation, you pull down this stuff, it's great through trim, you package it, and you start to lose a lot of these esters incredibly quickly. Have you looked at the delicacy of any of these compounds responsible for some of these fruity sweet notes?
So not specifically to these sweet notes over time. In our first publication, we did look at sulfur compounds and how they age, one, how it forms throughout the growth cycle, where it forms during cure, and then how it lasts throughout packaging. We saw with Prunal mercaptan, which is responsible for the gassy aroma, that really starts peaking around right as you're finishing cure. But then a week and a half after packaging, depending on your packaging scenario, you can lose 50% of those aroma compounds by weight. So packaging plays a huge role on why these things are either retained and delivered to the end consumer or lost, right? And also this is just goes to highlight how important curing is, storage, properly treating the plant, because these are delicate compounds. Esters are more volatile. Not all of them, but a large portion of them, like ethyl butyrate, is as volatile or more volatile than a lot of monoterpenes. So if a lot of these monoterpenes are being lost in the first 30 days, so are these fruity estery notes. But you can help mitigate that by proper packaging scenarios. If you're using a mild, think of your classical sandwich baggies, right, from the traditional days, the legacy days, it's not helping protect the material at all. It's probably going to last a couple of days before it's fully dried out and you lose everything. Whereas if you have a really well built out packaging solution, because you have, put a lot of time and effort in growing and curing that cannabis, if you can get in a nice packaging scenario, you can help protect it to that end consumer.
Hey, TJ. When we talk about stability, have you looked at the stability of the various compounds and even worked with some of the manufacturers to look at the stability from a flavor perspective and even a tox perspective?
So from a flavor perspective, it's tough on flower for sure. When you have extracts, one of the benefits are you have an ideal storage. One, you have more by weight. So you have more that can be lost before, percentage-wise, that can be lost before it has a specific aromatic, noticeable difference to the end consumer, right? And it's also in an ideal matrix. It can be stored in a fridge or a freezer to help protect those compounds. But if we looked at the toxicology of, say, one rosin or one flower, not really. We've looked at specifically compounds of interest and what those toxicological components may be, because obviously we're using Abstracts is obviously a terpene flavor house and a cannabis extraction company, so we care to see, one, what makes it smell and taste the way it does, but how do we do this in a safe or a more acceptable, meaningful fashion, right? And so we're not delivering anything negative to our customers and end consumers. But as far as how rosin ages on a shelf, not necessarily, at least not in the context of this.
All right. If there are no other questions, yeah.